Hello everyone, it is my great honor to share our research findings with you today. Non-human primates are facing a survival crisis. About 75% of species are declining because of habitat loss driven by human activities. Traditional conservation biology emphasizes three levels of biodiversity: genes, species, and ecosystems. In our study, we highlight a fourth level: the diversity and composition of biotic microbes, which play a vital role in host nutrition, health, and local adaptation. François langurs and white-headed langurs are ideal models for exploring the impacts of human disturbance and dietary niches. They live in fragile karst limestone forests and depend on microbes to digest high-fiber, oxalate-rich leaves. We collected fecal samples from wild populations in Guangxi and Guizhou, as well as from captive zoo populations, forming a gradient of human disturbance. Using PacBio full-length 16S RNA sequencing and deep metagenomics, we reconstructed 1,199 high-quality genomes and integrated nutritional, behavioral, and GIS-based landscape analyses. From 237 fecal samples, we found significant differences in gut microbiome composition. Dominant phyla included firmicutes, bacteroidetes, proteobacteria, and varicomicrobia. Each population showed unique enriched taxa, for example, Salvibrio in Guangxi wild langurs, Treponema in Guizhou wild langurs, and Presbytis in captive langurs. Environmental microbes also contributed to gut communities. The core species Pandaria noribergensis entered through food and water and showed seasonal variation. Importantly, langurs living near villages in Guizhou shared more similar microbiomes with captive populations, suggesting that human disturbance may drive microbial humanization. We revealed a clear gradient of anthropogenic impact, from Guangxi wild groups to zoo populations. With increasing disturbance, human-related taxa such as Lachnospiraceae, Bacteroideals S24-7, Presbytis, and Spirochaetaceae became more abundant. Treponema, Presbytella, and bacteroids were enriched under disturbance, likely linked to high sugar, low-fiber diets such as corn and sweet potatoes. Some treponema may originate from humans or livestock, raising potential health risks. We discovered strong microbial capacity for cellulose degradation, which is essential for leaf-eating adaptation. From 47 metagenomes, we reconstructed 1,199 NGs and identified widespread GH5 enzymes. In total, 177 NGs carried 2 key cellulases, and 6 super NGs carried all 3, mainly belonging to Salvibrio. These were enriched in the least disturbed Guangxi groups, matching their high fiber intake of up to 20%. We further identified 167 NGs involved in degrading dietary polysaccharides such as starch, inulin, and pectin. Their abundance increased with human disturbance, peaking in captive langurs. Human-related families like Lachnospiraceae and Bacteroidaceae were strongly enriched. Akkermansia mucinifera was higher in captivity, reflecting a shift to mucin utilization under low-fiber diets. 6 methanogen NGs, including Methanobrevibacter smithii, facilitated fermentation and SCFA production, enhancing host energy harvest. We also examined oxalate metabolism. Among 1,199 MAGs, 39 carried key oxalate-degrading genes, but dominant taxa varied among groups. Otococcus and Oxalocibacterium were enriched in Guangxi wild langurs, while Mirabaculaceae dominated in captive groups. These shifts suggest that both diet and human disturbance shape oxalate-degrading functions, reflecting microbial plasticity and host adaptation. In summary, different langur populations harbor distinct gut microbiomes and functional capacities. With increasing human disturbance, human-associated taxa became enriched, showing strong anthropogenic influence. At the same time, microbial functions such as polysaccharide degradation, oxalate metabolism, and mucin utilization provide active solutions for langurs living in karst limestone forests. We emphasize that symbiotic microbial diversity should be considered as a fourth level of biodiversity in wildlife conservation.